Another video of Clinical Biochemistry Unit 3. Let's dive. Definition of biomolecules. Biomolecules are organic molecules that are involved in the structure, function, and regulation of living organisms and cells. These molecules include carbohydrates, proteins, lipids, and nucleic acids, which play essential roles in cellular processes and life functions. Classification and metabolism of biomolecules. 1. Carbohydrates. Definition: carbohydrates are organic compounds made of carbon, hydrogen, and oxygen, with the general formula CN, H2O, NC_N, H_2O, NCN, H2O, N. They serve as a primary energy source for living organisms. Classification. Monosaccharides, simple sugars like glucose, fructose, and galactose. Disaccharides, composed of two monosaccharides, example sucrose, glucose plus fructose, and lactose, glucose plus galactose. Polysaccharides, complex carbohydrates, such as starch, glycogen, and cellulose. Metabolism. Glycolysis, the breakdown of glucose into pyruvate, producing ADP. Gluconeogenesis, the synthesis of glucose from non-carbohydrate precursors. Glycogen metabolism, involves glycogenesis, formation of glycogen, and glycogenolysis, breakdown of glycogen. 2. Proteins. Definition: proteins are made of amino acids, which are the building blocks of protein chains. They serve in structural, enzymatic, transport, and regulatory functions. Classification. Simple proteins, only composed of amino acids, example albumins and globulins. Conjugated proteins, contain non-amino acid components, such as glycoproteins and lipoproteins. Metabolism. Protein synthesis, involves the translation of messenger RNA, mRNA, into polypeptide chains. Proteolysis, breakdown of proteins into amino acids, which can be used for energy or synthesized into other proteins. Urea cycle, the process in the liver where toxic ammonia from protein metabolism is converted into urea for excretion. 3. Lipids. Definition: lipids are hydrophobic or amphipathic molecules that include fats, oils, and phospholipids. They are important for energy storage, membrane structure, and signaling. Classification. Fatty acids, saturated, no double bonds, or unsaturated, with one or more double bonds. Triglycerides, composed of 3 fatty acids and a glycerol backbone, serve as energy storage. Phospholipids, contain fatty acids, glycerol, phosphate, and a polar group, crucial components of cell membranes. Steroids, lipid-based molecules like cholesterol and hormones such as estrogen and testosterone. Metabolism. Beta oxidation, breakdown of fatty acids into acetyl-CoA, which enters the citric acid cycle to produce ADP. Lipogenesis, the synthesis of fatty acids from acetyl-CoA and glycerol. Ketogenesis, formation of ketone bodies from fatty acids, used as an alternative energy source during starvation. Each biomolecule has unique roles, and their metabolism is interconnected to maintain cellular energy and function. 1. Which of the following is the most common monosaccharide in human metabolism? A. Ribose. B. Fructose. C. Glucose. D. Galactose. Answer: C. 2. Which enzyme is responsible for the breakdown of glycogen to glucose 1-phosphate during glycogenolysis? A. Glycogen synthase. B. Glycogen phosphorylase. C. Hexokinase. D. Phosphofructokinase. Answer: B. 3. In glycolysis, which molecule is formed directly from the cleavage of fructose 1,6-bisphosphate? A. Glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. B. Glucose 6-phosphate. C. Pyruvate. D. Acetyl-CoA. Answer: A. 4. What is the net ADP yield from one molecule of glucose in anaerobic glycolysis? A. 2 ADP. B. 4 ADP. C. 32 ADP. D. 38 ADP. Answer: A. 5. Which of the following is not a polysaccharide? A. Glycogen. B. Starch. C. Cellulose. D. Glucose. Answer: D. 6. Which of the following best describes the primary structure of a protein? A. The folding of the polypeptide chain into a specific 3D shape. B. The sequence of amino acids linked by peptide bonds. C. The interaction of multiple polypeptide chains. D. The presence of secondary structural motifs like alpha helices and beta sheets. Answer: B. 7. What is the role of peptidyltransferase in protein synthesis? A. To separate the mRNA from the ribosome. B. To catalyze the formation of peptide bonds between amino acids. C. To add a cap to the mRNA. D. To release the newly synthesized protein. Answer: B. 8. Which of the following amino acids is essential and cannot be synthesized by the human body? A. Alanine. B. Glutamine. C. Leucine. D. Serine. Answer: C. 9. What is the function of the urea cycle? A. To synthesize amino acids. B. To convert ammonia into urea for excretion. C. To produce ADP from amino acid degradation. D. To synthesize proteins from amino acids. Answer: B. 10. Which of the following modifications typically occurs during post-translational processing of proteins? A. Phosphorylation. B. Transcription. C. Splicing. D. Replication. Answer: A. 11. Which of the following is not a characteristic of phospholipids? A. Amphipathic properties. B. Composed of fatty acids and glycerol. C. Found in the cell membrane. D. Used primarily for energy storage. Answer: D. 12. Which enzyme catalyzes the breakdown of triglycerides into glycerol and free fatty acids? A. Lipase. B. Amylase. C. Phospholipase. D. Protease. Answer: A. 13. What is the product of beta oxidation of fatty acids? A. Acetyl-CoA. B. Glucose. C. Glycerol. D. Pyruvate. Answer: A. 14. Which of the following lipids are primarily involved in the formation of biological membranes? A. Triglycerides. B. Phospholipids. C. Cholesterol. D. Fatty acids. Answer: B. 15. Which of the following is the main function of cholesterol in the body? A. Provide energy through beta oxidation. B. Form structural components of membranes and synthesize hormones. C. Transport fatty acids. D. Serve as an energy storage molecule. Answer: B. 16. Which pathway synthesizes glucose from non-carbohydrate precursors like lactate and glycerol? A. Glycolysis. B. Gluconeogenesis. C. Citric acid cycle. D. Pentose phosphate pathway. Answer: B. 17. In protein metabolism, the deamination of amino acids results in the production of which substance? A. Acetyl-CoA. B. Ammonia. C. Pyruvate. D. Glucose. Answer: B. 18. Which of the following is true about lipogenesis? A. It is the process of converting glucose into glycogen. B. It occurs in the mitochondria. C. It leads to the synthesis of fatty acids and triglycerides. D. It produces ADP and NADP as byproducts. Answer: C. 19. Which enzyme is involved in the final step of glycogen synthesis? A. Glycogen synthase. B. Glycogen phosphorylase. C. Hexokinase. D. Phosphofructokinase. Answer: A. Which of the following is not a product of the citric acid cycle? A. NADH. B. FADH2. C. ATP. D. Glucose. Answer: D. Thank you for watching.